And spinny ball, spinny ball. Okay, here we go. Now that I've laid some groundwork for just talking about parasites in general, we can start working through the major protist taxa, or the major parasite taxa, and we'll start with the protista. And we'll go back to the Swedish botanist, Carl Linnaeus. You might remember his name. He devised binomial nomenclature. He devised the ranked hierarchical system of classification, right? Kingdom, phylum, class, order, family, genus, species. King Paul came over from Germany swiftly, or however you learn to remember it. And he had originally, back in, gosh, now I suddenly can't remember what the official starting date is. I think it's 1763. He classified all living things into two kingdoms, Animalia and Vegetabilia. He'd actually tried to throw in the mineral kingdom as well, but he ended up jettisoning that. It didn't work so well. And we now call Vegetabilia the plantae. Everything was animals and plants. This was actually what I learned in the textbooks in dear old Acadiana High School in Scott, Louisiana, home of the Wreckin' Rams, back in 84, sorry. Animals moved and ate, plants didn't move, and they made their own food. Okay, sounds great. But as the light microscope started getting better, hi, come in if you want. Okay. The technology to make light microscopes got quite a bit better in the 19th century, and it started becoming increasingly obvious that some organisms just didn't fit either group very well. This was an early microscopist named Ehrenberg. Don't worry, by the way, about memorizing dates. Let's just say he was one of the first to demonstrate that single-celled organisms were complete organisms in their own right. He showed that they had structure within the cells. He saw nuclei. He saw vacuoles and other organelles, and argued that even though they were just made of one cell, they had all of the basic physiological functions that you would expect to find in a larger and more complicated animal. They ate, they excreted, they respired, and so on. So that means we've got to do something with the critters that don't fit, like the protists that are green and also feed, for example, or the critters that aren't green but don't feed in the way that animals do. A guy by the name of Ernst Haeckel put everything that wasn't a plant or an animal into what he called the kingdom protista. His protista included fungi, and it also included prokaryotes. And I'll mention this very briefly. Lynn Margulis, who came up with this idea of serial endosymbiosis, came across an older paper by a guy by the name of Hogg, who had the same idea but put these organisms into a kingdom protoctista. Margulis always argued that protoctista was the right name for these, and you'll see this term if you ever read stuff written by her or by people closely associated with her. She wrote about protoctists an awful lot. So you might see it if you go on to do further reading, but outside of her own circle, the term never really caught on. People still say protist a lot. I know you could say that maybe they're protesting. Maybe it's a protest movement. Okay, I'm just imagining 23 of you rolling with laughter. By 1937, the basic distinction between prokaryotes and eukaryotes was worked out. So that splits off the bacteria and the archaea. And then a chap named Whitaker in 1969 took the fungi out of this protista and created the basic 5 kingdoms scheme. And the 5 kingdoms was what I was taught back in my undergraduate years, back in the days of hair metal, and what we taught in when I was a grad school instructor, and quite possibly what you learned. The 5 kingdoms scheme of prokaryotes or Monera, animals, plants, fungi, and protista. Well, I want to protist this scheme, maybe wave a protist sign, because the protista is a wastebasket. The protista is where you throw everything that isn't an animal, plant, or fungus, or every eukaryote that isn't. The defining characters of the protista are that it doesn't undergo extensive embryonic development, and there's not extensive cellular differentiation. You're grouping these critters together based on what they aren't, rather than what they are. An old friend of mine back in high school, with a sense of humor that's even more surreal than mine, used to make up jokes like, what does a bald eagle have in common with a brick wall? The answer, of course, is that neither one can drive a stick shift. Okay, to the extent that's funny at all, it's funny because bald eagles and brick walls are completely different except for the things that they don't do. And grouping them on the basis of what they don't have and can't do is silly. But that's what we do with the protista. The protista includes some very close relatives of true animals, true plants, and true fungi, and a whole screaming bunch that don't resemble any of those at all. So throwing everything in the protista is really masking the ridiculous amount of diversity that's really there. So as we define the protista, it's paraphyletic, meaning it includes some descendants of a common ancestor, but it doesn't include all. The last common ancestor of everything we've called the protista was also an ancestor of animals, plants, and fungi. This is a complete mess. Well, it's not, but the type is pretty small. I'm not expecting you to read this. This is a cladogram of eukaryotes based on a very big DNA data set. And this is pretty much all of the eukaryotes. There's a few that are hard to place that aren't on here, but this is representatives of all the major groups. That yellow box shows you where the animals are. All of the land plants are in the green box. Here, trying to make sure everybody's awake. And all of the fungi belong in the red box. And so we have some protists down here that are close to the animal kingdom. There's some that are close like that are close to the fungi. There's some protists up here that are close to the land plants. And there's a whole bunch that aren't especially close to any of them. They represent completely separate branches of the eukaryote domain. And so I still use the term protist when I'm teaching biology 1400, but what you initiates into the higher biology need to know, yes, hail thou who art Darwin, even thou who is Darwin. Sorry, higher biology. We need some rituals and secret handshake or something. Those of you that are initiated into the mysteries of the higher biology need to know that we don't really use kingdom Protista anymore, and eukaryotes are divided into several different groups that you might not be familiar with. We're not going to go over every last one of them. If you really want that, you can take me for invertebrate zoology, assuming they ever let me teach that again. They probably will. We go over all of them in invert zoo. In parasitology, we're just going to go over a sampling of them. And the current scheme that we've got uses 5 supergroups of eukaryotes. The Amoebozoa, which includes, amoebas. A supergroup that doesn't really have a proper Latin name. It's called SAR. And SAR stands for Stramenophiles. Stramenophiles includes lots of things, but among others, it includes brown algae and golden algae and the potato blights that nearly wiped out Ireland in 1847, and various others. There's lots of things in the Stramenophiles. The alveolata we'll talk about later, because there is a major group that's parasitic in the alveolata. The Rhizaria, which is a bunch of neat little amoeba things with shells that don't have a, none of those are parasites as far as I know. So we have parasites in the Amoebozoa, parasites in this alveolata group, which is a subdivision of SAR, and parasites in a group called the Excavata, which we'll be talking about in a week or two. Opisthocanta includes animals and fungi and those protists that are more closely related to them than to anybody else. Certainly there are lots of parasites in the Opisthocanta, including all of the animal taxa we'll talk about, as well as many fungi. And then plants, green algae, red algae, and an obscure but interesting group called the Glaucophytes belong into a larger group called the Archaeplastida. There aren't many parasitic Archaeplastida. There are a few parasitic plants. We're not going to talk about them just for reasons of space. And we're not going to talk about parasitic fungi. And some of those SAR groups don't have very many parasitic members. But we do want to focus on the Amoebozoa, the alveolites within SAR, and the Excavata are the major eukaryote groups that have parasites in them. And also, of course, the kingdom Animalia, which has, worms and stuff like that. And we define these groups by similarities in DNA sequences, but in many cases we can find similarities in structure as well. And we'll talk about those as we go. So we'll start marching through the parasites with the Amoebozoa. Many amoebas go here, but amoeboid shape has evolved several times. These things aren't classified together just because they are amoebas. If you don't know what an amoeba is, you should look under the microscope, and if you see one, you can sing, "Amoeba! I've just seen a protist amoeba." All right, I'll give you a few seconds to get over your breath being taken away by my vocal register. Right, amoebas don't have any fixed shape. They do have a tendency towards a particular shape and size. So some amoebas have many lobes sticking out from the main body of the cell that are called pseudopods. Others have few. Some, the lobes are broad, others they're narrow. Some amoebas will be in, let's say, the 500 micron range. Others might be 50 or less. So you can actually define different species of amoebas, despite the fact that amoebas don't have a permanently fixed shape. Amoebas move by amoeboid locomotion, and that means the cell will put out one or sometimes more extensions of the cell body called pseudopods, Greek for false feet, and the cytoplasm flows into the pseudopods. By the way, that's the same way that your white blood cells get around. Amoebas typically feed the same way your white blood cells feed, sending out a couple of pseudopods to surround something and engulf it and digest it. That's called phagocytosis, and your white cells do the same thing. So you might have seen that term before. And there's a classic free-living amoeba. This is one that I bring into my invertebrate zoology class, 4401. This particular one happens to be quite large. This is maybe 500 microns, possibly more. And the nucleus is kind of in the middle. If you can see, it's that little grainy thing. There's a vacuole next to it. There's a kind of tail end called the uroid at the left. And the amoeba is sending out multiple lobe-shaped extensions of itself. Those are the pseudopods. And in fact, you can see two of them that are reaching around and about to fuse and engulf a helpless green alga. That's phagocytosis. Okay, this amoeba does not bother anybody. These are free-living and perfectly harmless. And exactly how they move is still not 100% understood, but the basics are that amoeba can make their cytoplasm take on one of two different states called sol and gel. Sol is a liquid state, it flows easily, it's runny. Gel is much more firm, similar, maybe not quite as firm as Jell-O, but the same idea of a flexible but firm substance. And amoebas can transition parts of their cytoplasm from the sol state to the gel state by building cytoskeleton very quickly, forming polymers of actin, the protein actin. When cytoplasm is in the sol state, the actin is broken into small pieces and the stuff can flow very easily. So the inner cytoplasm in the sol state, we call it endoplasm, can flow forward. When it reaches the front of a pseudopod, it kind of fountains around and goes in all directions. And as it comes back, it locks into gel. So just back of the advancing tip of the pseudopod, this flowing sol turns back into gel. And if I can find a video, I'll post it. It's really neat. If you watch one of these move and you watch a pseudopod under the microscope, you'll see this kind of internal fountain of freely flowing grainy cytoplasm pushing its way forward, coming around, and then immediately locking into position as part of this gel. It's a lot easier for me to show than to describe, but it's pretty neat stuff. The outer cytoplasm, called the ectoplasm, locks into place in that gel state. And we think that the cytoskeleton squeezes from behind and forces that sol, that free-flowing stuff, forward. So we have contraction of actin and myosin, the same proteins as do the work in your muscle. Contraction at the rear, squeezing endoplasm forward. So that endoplasm turns into solid, flows forward, locks into position as gel, and then eventually is pushed to the back where it turns into solid again. And again, I'll try to remember to post a YouTube video of this because 10 seconds of that will make this a lot more clearer than my yelling. There are some amoebas that are facultative parasites. This one is extremely common in soil or freshwater. Typical size is maybe something like 25 microns. It's not that big. Acanthamoeba means spiny amoeba because it has much narrower thorn-shaped pseudopods rather than the big blobby ones of the one you just saw. And it's very hardy because when conditions are bad, it can secrete a wall around itself and form a cyst. So on the right, that is an acanthamoeba cyst that can resist drying out, can go without food. I'm not sure what the record for survival is, but it's at least several months that they can survive in the cyst state. And then if conditions get better, they'll just pop right out and go back to living their amoeba life. Most acanthamoeba live, just hang out in the soil and don't bother anybody. But if you get them in your eye, well, hopefully your tears would wash them out. But if you get them in, let's say, contaminated contact lens solution, especially if you have those extended wear contacts, that means you're not taking them out and cleaning them really well every night, brace yourself, you can get this. So this would be facultative. Acanthamoeba can live just fine on its own, but if you get it established in the cornea of your eye, you get pretty severe eye infection that could lead to blindness if it's not treated. Very rarely, they can get up your nose and into the brain and cause amoebic meningoencephalitis, although the most famous brain-eating amoeba actually belongs to a very different group, and we will have occasion to talk about them later. So acanthamoeba can be a facultative parasite, and it's a very good argument for, watching what you're using for contact lens solution very carefully, and you probably really should take them out every night. Acanthamoebas also tends to be packed full of bacterial symbionts, and I don't think we fully understand what they're doing in there, but some of these symbionts are capable of causing disease. One of them is Legionella, which causes Legionnaire's disease, a very severe lung infection. This is a cross-section through an acanthamoeba packed full of another bacterium that can cause lung disease. This is called parachlamydia. And acanthamoeba seems to be a natural reservoir host for these bacteria. They can hang out in the acanthamoeba cells and then go on to invade yours. Right. As for the true parasites that come up a lot in medicine and veterinary medicine, probably the best known is Entamoeba histolytica. Histolytica means it breaks down cells. This is the causative agent of amoebic dysentery. Enteron is Greek for intestine. Dysentery means bad intestine. So this is amoebas doing bad things to your intestine. And by bad things, I mean potentially bleeding ulcers, potentially perforating your intestine completely and starting to eat your liver, certainly severe diarrhea and symptoms like that. And the life cycle of these guys alternates between an active amoeba-like feeding form, a creeping amoeba called a trophozoite, and an inactive form that can pass from person to person, called a cyst. By the way, trophozoites, trophane is Greek for to feed. And zoon, of course, zoo means to live. So a trophozoite is a feeding, living thing. And we'll actually see that term used elsewhere. There's lots of protist parasites that have a stage that's called a trophozoite. So they'll alternate between trophozoites, which eat your intestines, and these inactive cysts, which is usually the stage that's transmitted from person to person. So the trophozoites colonize your intestinal lining. As they grow, some of these trophozoites form cysts, and the cysts pass out in feces. And you're going to see this term over and over and over again. They reach new hosts by what's called the fecal-oral route, which, don't think about it too hard, it's really gross, but that's what it's called. They reach new hosts who ingest feces of the old hosts. Usually that's not direct, at least not in humans. Cockroaches do it all the time. That's how our South American roaches in the lab get their symbionts, because they eat each other's poop. In humans, it's much more likely to be ingestion of food or water that's contaminated with feces. You can get this in areas that don't have the best water purification. You can get lots of things by the fecal-oral route if you're in a wilderness area and you drink water that hasn't been sterilized. You know, even water from a clear running mountain stream can have things in it that you don't want to get into your guts. You could get it if whoever prepares your food does not wash his or her hands very well. Kids may get parasites by the fecal-oral routes for reasons that I trust I don't have to spell out. So if something goes from feces to ingestion by another host, that's the fecal-oral route. There's lots of ways that it can play out. There is new evidence that it can be sexually transmitted, usually among gay men. There's no reason why other particular pairings and couplings could not transmit it as well. Trust I don't have to spell that out. Let's go. That's a trophozoite stained. This is usually diagnosed by taking a fecal smear from an infected person, smearing it out on a slide and dunking it into various stains. And this is what a trophozoite looks like. Unlike the big amoeba proteus that you saw earlier, or acanthamoeba, which has lots of pseudopods, typical entamoeba only has one. The nucleus is dead in the middle, and the nucleus typically has a darker spot in the center. So the nucleus will have kind of a dark outer border and a dark central spot. That dark central spot is called an endosome, and a lighter zone in between the two. So that thing that looks a little bit like an eye is the nucleus, and that eye with a dot in the center of it is very typical of this group. The dark spots are food vacuoles filled with red blood cells that the amoeba has engulfed. And you can't see this here, but entamoeba has lost its mitochondria. It used to have them. We've got DNA sequences in the nucleus that were transferred from ancient mitochondria, so its ancestors had them. But entamoeba itself does not have these anymore. And as such, it can only live in anoxic environments, like your gut, which is pretty anoxic in your large intestine. %90 of all infections are asymptomatic, and they may actually be caused by not true Entamoeba histolytica, but a different species called Entamoeba dispar. 10% of the time, though, the amoebas start eroding your intestinal lining. And this is what these look like under endoscopy using a sigmoidoscope or something like that. You can see, especially here on the left, those white patches are areas where the intestinal mucous membrane is just getting eroded away. Over in the picture on the right, you can see some areas that are oozing a bit of blood, especially up at about your 10 o'clock right there. Those amoebas are becoming invasive. They start inflaming the intestinal wall. This can lead to permanent intestinal ulcers. And in really bad cases, the ulcers may perforate and the amoebas can crawl out. They can enter the bloodstream or they can end up in other organs. I don't know why I didn't get rid of that. They can end up in your liver. They can end up in your brain. They can end up in all sorts of places where you really don't want wild amoebas. That's a liver with a amoebic abscess in the middle of it. They cause these giant abscesses loaded with, I guess they've washed it out. They're supposed to contain this pus that looks like chocolate sauce. Sorry, I probably put a couple of you off your feed. I hope you weren't eating breakfast, but this is parasitology. This is kind of a gross course. They can also end up in your lungs, in your skin, or in your brain. That's a brain abscess due to Entamoeba histolytica. Right? Not something you want, amoebas in your brain. Yep, you want to get treatment for this. And when the treatment is through and the abscess is completely gone, you can say, adios, amoebas. It's a joke. You don't have to write that down. That's a joke. Okay, great. We haven't ever caught Entamoeba having sex. We know it's got genes that in other organisms are linked to the ability to undergo meiosis. But nobody's ever seen direct sexual reproduction, probably because I don't know how easy it is to culture these guys outside a human host. But I'm guessing it's not particularly easy. It may just be that we've never seen sexual reproduction happening because we haven't figured out how to create the conditions that will cause it to happen when we study these in the lab. Okay, in most species, some of those trophozoites will form cysts. During cyst formation, the nucleus may divide and create different numbers of nuclei, and that's species specific. Entamoeba histolytica is your bad guy, and that's the one at the top right of this slide, and you can see 4 nuclei per cell. And remember, each of those nuclei has that darker rim and that darker central spot called the endosome, and it's lighter in the middle. So that kind of bullseye nucleus, those of you that want to be doctors or veterinarians might actually see this down the road. So the one at the top left is Entamoeba histolytica, 4 nuclei per cyst. Down below, Entamoeba coli is harmless. It's a commensal. And you can't count them all because they're not all in focus at the same time. But you can see it's got more than 4, and in fact, it's got 8. And then there are some species that only have 1 nucleus per cyst, and some might have 2. So this is often important for identifying whether you've got a harmless or a pathogenic species living in your guts. The cyst also contained this body called a chromatoidal rod. It contains protein. It seems to be a food reserve. And if you stain them, over there on the right, that's a stained Entamoeba cyst. And you can see this big, kind of vaguely oval-shaped bar in the middle that stains very strongly. That's the chromatoidal rod. There's an unstained one on the left, and the rod is harder to see, but you can still make it out. So this is what you were looking for. Now, I've actually got in lab some slides of pre-made fecal smears that are supposed to have entamoeba in them. I've personally never had much luck in finding them. It can be kind of a needle in a haystack thing. But I might have an excuse to take those out and make you folks look at them when you come in for lab. You are going to get some work done during your formal lab meetings using the practice slide collection that we've got. So you may end up having to hunt for these things. And yeah, here's a few species. Histolytica is pathogenic in humans, sometimes dogs and cats. Entamoeba coli and dispar are harmless. There's a third species that lives in the mouth, Entamoeba gingivalis, which is also supposed to be a harmless symbiont, commensal like this. There's at least one species that just lives in sewage, but it's free-living. It's not parasitic at all, Entamoeba mushkovskii. And then you'll find others that are commensals, parasites in pigs, horses, ducks. Entamoeba invadens is a parasite of lizards, which might not seem that interesting, but it's now used as a model system for studying human infections. You can infect a bunch of lizards and then do experiments on them. And there's lots of others. You can see from the scale bar, they're not that big. 10 microns makes them kind of annoying to work with. And I'm sure there are lots more species out there that nobody has really bothered to go looking for. And as you see, many of them are commensal, and they're not ever likely to cause symptoms that would make you want to go looking for them. So this is a group that's probably a lot more diverse than we suspect. And there's a few related genera. Endamoeba is one that I have seen in our South American roaches before. I'm not sure how common it is, but A, it's very small, and B, since amoebas move very slowly, you're likely to overlook them if you're panning around quickly. And they're also transparent, meaning you would have to find a very small blob and watch it for a couple of minutes to see if it moves. And not everybody has the time or the patience for that. So I have had students find this in cockroaches before. It's probably very common, we just don't often see it. Because most of you are spending your time counting gregarines or things like that. But you can find these guys in roaches, probably commensal. I don't think they cause any harm. Another harmless one that you'll find in humans is iodamoeba. So-called because it stains brown with iodine, which is what you see there on the right. Another one in humans called endolimax may cause human disease, but again, it hasn't been very well studied. So I think we're actually finishing up early, but that's what I had to say about parasitic Amoebozoa. And we'll move on to, I think, the alveolata on Wednesday. So, by the power vested in me by the state of Arkansas, I'm going to go ahead and stop recording.